Hello students, welcome back to a new video of Sunanda's tutorial. So in this video we will discuss about phospholipase C or PLC pathway. Okay. So this is a structure of this pathway. So at first, what is phospholipase C pathway? It is a very crucial signaling pathway where phospholipase C enzymes hydrolyze phosphatidylinositol 4,5-bisphosphate PIP2 into diacylglycerol and inositol 1,4,5 triphosphate, which is IP3. So basically phosphatidylinositol 4,5-bisphosphate will be cleaved into two parts. One is diacylglycerol and second one is inositol 1,4,5 triphosphate IP3 by the PLC enzyme. Okay. Which this IP3 then act as secondary messenger to regulate intracellular calcium release from endoplasmic reticulum and activate protein kinase C. So the first step is activation of phospholipase C. So it is activated by various extracellular stimuli including hormone, growth factor, neurotransmitters, which bind to cell surface receptors. These receptors can be G protein coupled receptor, one is GPCR, or tyrosine kinase receptor, but mostly it is GPCR or G protein coupled receptor. So at first signal bind with the G protein coupled receptor. So next, after the binding, phospholipase C is activated by the binding of these ligands to their respective receptors. Then hydrolysis of PIP2. Once activated after binding with GPCR, the PLC or phospholipase C, which is present in the cell membrane, hydrolyzes PIP2, a membrane lipid, into two secondary messenger. One is DAG, second one is IP3. DAG remain in the membrane while IP3 release into the cytoplasm. So basically in this pathway, at first one ligand is attached to the GPCR receptor. Next it gets activated. Then it activates another enzyme PLC, phospholipase C. Phospholipase C can cause the cleaving of PIP2 or hydrolyzation of PIP2 into DAG, diacylglycerol, and IP3. Okay. DAG remain in the cell membrane and IP3 again release into the cytoplasm. Action of this secondary messenger IP3 and DAG. So what IP3 do? It binds to IP3 receptor. It binds to IP3 receptors on the endoplasmic reticulum and triggers the release of stored calcium ion into the cytoplasm. Next, what DAG or diacylglycerol do? It activates the protein kinase C or PKC, which phosphorylates various other proteins, leading to a cascade of cellular response. Calcium ions released by IP3 also contribute to protein kinase C activation. Okay. So basically this is the pathway. At first signal from external environment bind with the GPCR or receptor, then it activate the GPCR receptor, then it activate the phospholipase C. Phospholipase C cause the cleavage of PIP2 into DAG and IP3. DAG remain in the cell membrane and causing the activation of protein kinase C. IP3 again bind with the surface of endoplasmic reticulum, causing the exit of calcium ion outside, that means into the cytoplasm. What are the downstream effects? So downstream effects include the phospholipase C pathway plays a role in various cellular processes. What are those? One is calcium signaling, because IP3 mediated calcium release is crucial for many cellular function, including including muscles contraction, neurotransmitter release, and cell signaling, other cell signaling pathway. Next is protein kinase C activation. Next, cell proliferation and differentiation, stress response. So tumor development and progression, dysregulation of this pathway, phospholipase C pathway, can contribute to tumor development and progression. So it may lead to the development of tumor, and the development of tumor, progression of tumor, rapid division of the cells. So these are different downstream effects, positive as well as negative effects of this pathway. So it is overall about the phospholipase C pathway, phospholipase C signaling pathway.